Most of us often tend to think that critical thinking is negative or destructive thinking, but that's not the case. The goal is to get as much near as possible to the truth. So you need systematic answers without accepting everything at face value. This video will explain the concepts of critical thinking with a suitable example and how this framework of critical thinking could be used in your work presentations or while preparing research proposals or even writing review articles. So let's dive in. Critical thinking has three core processes. Number one, description. Number two, analysis. And number three, evaluation. Let's take an example of blood disorders and their treatment modalities as an example. So, how could you critically analyze the treatment options available and make a presentation or present your perspective based on the available data? So, let's start with description. In general, the questions under description are, let's say if you're doing a case study, then you'll ask the following questions. Number one, what is the situation and who is affected or even involved? Number two, when did it happen? Number three, where did it take place? So you first need to describe the problem or the current status of the problem. So, in this example, let's get to know the background of beta thalassemia. So, you've collected information from different articles and give the following description. So, first, we describe the molecular basics of beta thalassemia and how it affects the body. In this case, we'd say it is caused by the mutations in the hemoglobin beta subunit gene and it results in reduced beta globin synthesis. And then we would continue with the molecular basics of the disorder. Next, we focus on the treatment modalities that are available till now, starting from the traditional approaches. So, first, we talk about the traditional treatment for beta thalassemia, which is allogenic bone marrow transplantation from a completely matched donor. But the problem with that method is that there are limited number of human leukocyte antigen matched donors. Then the additional problems are the patients need long-term use of immunosuppressive drugs and there is a high risk of immunological complications. So this approach has limited application. Another approach uses transfusions. So patients require frequent blood transfusions and chelation therapy to restrict iron deposition. But the problem with this method is that despite improvements in transfusion practices and chelation treatment, you definitely need newer therapeutic strategies such as nanomedicine or something like gene editing. Then we focus on molecular medicine. What are the target sites that you could use? Here we talk about the different approaches used. So, one of the approaches is the epigenetic approach that is used to reactivate fetal gamma globin. And research has shown that increased synthesis of this fetal hemoglobin can significantly decrease the intensity of thalassemia's major symptoms. So, some studies have been done using chemical compounds such as the 5-azaz-C, which is a demethylating agent, and they've seen that fetal hemoglobin reactivation actually helps. The next step is how this can be done as a targeted gene therapy. So here we focus on gene editing tools. So current gene editing tools, they are very powerful tools and hold great promise in treating beta thalassemia. So these tools include the transcription activator like effector nucleases, zinc finger nucleases, and the CRISPR-Cas nucleases. After this, we focus on what are the target sites. Once we've selected the target site, which in this case is fetal hemoglobin, we look at what are the factors which suppress the expression of this hemoglobin. So, in short, we focus on regulators of fetal hemoglobin expression. So, what are the molecules that regulate the expression of fetal hemoglobin? And studies have found that BCL11A has emerged as a major repressor of fetal gamma globin gene expression in the adults and represents a possible target for therapeutic intervention. With all this information, now we have a background in place for this topic on beta thalassemia. The next phase of critical thinking is the analysis phase. Once we have the description ready, we now analyze the information that we know now. In general, the analysis part consists of the following questions that you can ask when you are critically analyzing the concept. Let's start with some how questions. Number 1, how did it occur? If it's a case study. Number 2, how does it work in theory and practice? Number 3, how does one factor affect another? Number 4, how do parts fit in the whole? In the example of beta thalassemia, you could focus on these how questions. For example, how does gene editing work? The principle of gene editing and how somatic gene editing is performed. Next, you could explain how gene editing is better than all the therapies that we've seen so far, and also look at how the non-homologous end joining mechanism of gene editing and the homology directed repair mechanisms compare here. Are there different kind of studies available? Then you could ask, how does fetal hemoglobin production affect the whole system? Then in the next stage, you will focus on the why questions. Some questions that you ask here are: number 1, why did this occur? Number 2, why was that done? Number 3, why this argument, or why this theory, suggestion, or solution? And finally, number 4, why not something else? So in this section, you could ask questions such as: number 1, why CRISPR-Cas is the tool for gene editing compared to Xerifins and Talents? And then you could ask, why is fetal hemoglobin production better than correcting the gene mutation itself? Or number 3, why is BCL11A used as a target for knockout? Are there other options as well? So once you find out answers for these questions, then you could focus on the what if. Some of the what if questions are: number 1, what if it's wrong? Or what if there is a problem? Then number 2, is there any other alternative? And number 3, what if the factor is added or removed? Here in this example, you could focus on some of the what if questions such as: number 1, what if NHEJ mechanism is harmful and lead to on-target deletions? Number 2, is there any other alternative other than the NHEJ repair mechanism? What if you could use base editing or prime editing instead? Will they work? Are there publications in this direction? Will the outcomes be better? And the final question would be: what if one or both BCL11A genes were knocked out? Is there any kind of problem? Once you keep on asking questions in these directions and expand your thinking, you get a clearer perspective, a better understanding of the topic. Another important point to consider here is, while evaluating your sources, you should look at the following aspects. Number 1, you should look at the author. Is the author's article peer reviewed, or is he or she well known in the field? The next point to be considered is objectivity. Are the articles biased or balanced? Number 3, timeline. Is the article old or recent? And number 4 is relevance. Is the information complex or basic? Or is it broad or even narrow? Once you've analyzed and gone through research articles which answer your questions in the analysis section, then you move on to the evaluation section. In general, the evaluation part consists of the following questions that you can ask when you're evaluating the topic. So let's look at some of the questions that you could ask while evaluating the concept. Number 1, what does this mean? Number 2, why is it significant? Number 3, is the data convincing? Why or why not? Number 4, so what if it's successful or not successful? Number 5, how does it meet the criteria? So in the example of beta thalassemia, you could focus on what the current results from the clinical data are and how significant they are. Then you could check out why or why not the data is convincing and what is the criteria behind it. When is it okay to use non-homologous end joining based approach, or when do we need a homology directed repair based approach. Now, let's focus on what next questions. Number 1, is it transferable? Next is number 2, where can this be applied? Number 3, what can we learn from it? Number 4, what do we need to do now? Once you're done with these questions, you could ask the following questions in this example of beta thalassemia. What is next in the treatment of such blood disorders? Can it be applied to other blood disorders as well? Has anyone worked on base editing and prime editing for blood disorders? Is it time to focus more on homology directed repair based strategies? What can we learn about these technologies and the targets used for treatment of beta thalassemia? Once you've evaluated your findings, congratulations, you've critically analyzed that topic. Now you can make a fresh presentation with your own perspective based on the critical analysis that you've done and start building on your proposal or any piece of work that you wish to explore further. And so these are the steps for critical thinking. So now over to you. How do you intend to use critical thinking in your work?